Hi everyone, it's Mark here. This is just a little demonstration video to let you know how to complete the latest of our online labs. This one is called DNA, RNA, Protein, SNORKS, and it was adapted from a little exercise created by biologycorner.com. So what it's going to be is a little simulation where you examine the DNA sequence of a fictitious organism known as the SNORK. Now, the SNORKs were discovered on planet DNA in a distant solar system. They're much less complex than us. They only have one chromosome, and on that chromosome, they have 10 genes. So your job is going to be to analyze those genes and determine what traits the organism has, and then draw that organism and color it in. It could be as creative as you like, and then you'll eventually submit that drawing to Moodle. So the actual sequences, the gene sequences of SNORKs are much smaller than human gene sequences. And each gene has two versions that result in a different trait being expressed in the SNORK. So what you're going to receive are some DNA samples taken from volunteer SNORKs. And don't worry, no SNORKs were harmed in this process. And your job is going to be to translate that DNA sequence into RNA and then translate that RNA into a polypeptide and then determine the phenotype, i.e. what it looks like based on the amino acid sequence. Now, all this will become clear in a second when I show you two sequences that I'll work through. The only thing to really remember is that the sequence AUG is a start codon and signifies the beginning of each gene. UAA is one of the three stop codons and signifies the end of a gene. For simplicity, you've only been given the sequence of the DNA coding strand, so it'll only be single-stranded and not double-stranded. So as I said, I'm going to demonstrate how to do this. And then what you'll do is you'll go to Moodle and you'll select the package. So it'll be a Word document, and you have to select the one that corresponds to the range that contains the first letter of your family name. So then you'll go ahead and do your transcription and translation, figure out the traits, draw your SNORK, take a picture of it, and then you can go to Moodle to fill in all your answers on the Moodle quiz. And remember to select the one that corresponds to the range that your first letter of your family name comes in. So without further ado, I'll demonstrate how to do two different DNA sequences here. So as I mentioned, you're only getting the DNA coding strand. So normally this would be double-stranded, but the coding strand is what's complementary to the mRNA. So we've decided to take that. So all you do to transcribe it is that you have to transcribe, remember that T is paired with A when it's mRNA, G is paired with C, C with G, and remember, because RNA doesn't have thymine, it has uracil, that means A is paired with U. So to do this one, it's going to be A, C, A, U, G, G, U, C, A, G, C. You get the drift. A, U, A, C, G, G, C. So that's the sequence of the mRNA that would be produced. So that would be your first answer. Now, to get the polypeptide, you have to now act like the ribosome. So let's just change color. And you don't just simply start at the very start. There's a very specific start sequence that the ribosome has to recognize, and it's this. It's called the AUG. So the way things are done, as you've seen in classes, is that mRNA is read in groups of 3 nucleotides. So these are called triplets or codons. So what you do is you simply take the mRNA, once you've found the start codon, and then you divide it into 3s like this until you get to the stop codon. So as I mentioned, the start codon is AUG, the stop codon is UAA. So what we have are 3 codons here in the middle that we're going to now translate. So basically what the AUG does is it sets what's called the reading frame to know which group of threes to take each of the nucleotides in. So let's just see what we have here. So GUC, if I go to this little translation table here, you have to go GUC. So we start with the first letter is on this side, so G. Second letter is U, so it's going to be in this box somewhere. C. So the first amino acid on our polypeptide is going to be val or valine. So our polypeptide is going to be val. And almost every single polypeptide starts with AUG, which is methionine, but we're just going to skip this for simplicity for this particular exercise. The next codon is AGC. So we'll go back to our table again. A and then G down here, AGC, is ser or serine, like so. And the final one in this particular sequence is AAA. So again, first letter A, second letter A, it's going to be in this box. And there it is, it's lys or lysine. Okay, so now we have our polypeptide, valine, serine, lysine, and then we can go to this little table here and see where we see valine, serine, lysine. And we can see that here it's gene 1, and it means that the phenotype of this particular SNORK is hairy. So our phenotype is going to be hairy, like that. It's very simple, and then we simply start again with the second gene. So I'll show you that once more. Again, we have to transcribe this. So RNA polymerase would be the enzyme doing this, and it splits the two strands of DNA apart so that it can read the DNA, reading off the coding strand. So this one's going to be C, U, A, U, G, U, A, C, 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 G, A, G, A, G, A, U, A. This is a long one. U, A, C, C. Okay, so I'm almost as quick as RNA polymerase. So that's the answer to our mRNA. So that's our answer there. And now we have to go and get the polypeptide. So again, don't forget, don't just start making threes. You have to set the reading frame, and it's set by the start codon. So there's our start there. And so then it's read in threes like this. And there's UAA, so there's our stop codon. So we know that this one's going to be 1, 2, 3, 4, 5 amino acids long. So let's do that. The first one, UAC. UAC, I'll go a little bit quicker. Tyrosine. So I'll write that down here. CCC, proline. GAA. It's glutamate or glutamic acid. GAG. It's glutamate again, or glue. And finally, AAA. There it is again, AAA, lysine again. So now we have a polypeptide with 5 amino acids in, so that would be our answer for gene 2. It's tyr, pro, glu, lys. Tyr, pro, glu, lys. So this means our SNORK here, and this is, yours is going to be different from this because mine is a different SNORK. You're going to see that this one's phenotype is plump. So our phenotype here is plump, like that. And so you just go through them gene by gene. There's going to be 10 genes. All you have to do each time is transcribe into the mRNA using the the sequence code that I gave you at the top, and then remember to look for the start codon and kind of delineate your reading frame of 3 bases, nucleotides, to set each codon, and then read the codons off the chart that in the way that I just showed you to get the polypeptide and then get the phenotype. And it's as simple as that. And when you get to Moodle, you're simply going to take the document range that corresponds to your family name. So mine, Stocksley, begins with an S. So if I was to do this as a student, I would choose this one because S comes in between O and Z. So make sure and choose that. And then when you do the entire exercise, you can write it all out on a piece of paper, and then you'll have to come and do the quiz that corresponds to the correct range as well. If you go into one of the other quizzes, then all your answers are going to be wrong simply because they're different types of SNORKs. So once you get into the quiz, you'll end up with something that looks a bit like this. And what it's asking you is go gene by gene. So here's gene 1, and to enter the mRNA sequence without spaces. So this is really important. Make sure not to put spaces in, otherwise it's going to mark you as wrong. So this is going to be where you would type it out in here. Then there's a drag and drop to do your amino acid sequence. So you can simply drag and drop the correct amino acids based on their abbreviations. And then finally, you write down the phenotype of each of the sequences. So for sequence 1, and it's really important to enter it as it's written on the worksheet. The exact same spelling, the exact same word order, because otherwise Moodle won't pick it up as being correct. So make sure and type it in. So if it says, for instance, blue pigment, hair, skin, then it has to be typed out exactly as it is here for Moodle to be able to recognize it as being correct. The final thing that you'll do is you'll draw your SNORK, color it in, take a picture with your phone, and then send that picture it's the final question in each of these quizzes is to attach your file. So you'll attach that to Moodle, and then it'll accept it, and we can look at your wonderfully created SNORK and see how artistic you are. So good luck with that. If you have any questions or you have any problems or concerns with that, then please send me a Mio, and I'll be very happy to get back to you and help clear up any confusion that you might have. So good luck and enjoy transcribing, translating, and drawing your SNORKs. Bye.